Welcome to agrobacterium-mediated transformation of onion epidermis for transient assays with REX. This particular method transiently expresses GFP on onion epidermal cells using agrobacterium. So this study can be used in order to analyze the expression of genes or promoter in a transient analysis. The materials required are agrobacterium, which harbors the GFP fused with your gene of interest, or a promoter. So the first step in this particular agrobacterium transformation of onion epidermal cells is the preparation of agrobacterium culture. So from a single cell, we try to inoculate or prepare a mother culture. So we take a single colony from the agrobacterium, which is harboring the GFP protein fused to the gene of interest or the promoter under investigation, and inoculated in a 5 ml LB broth. The broth is pre-inoculated with kanamycin selection as well as rifampicin selection. Kanamycin selection is 50 microgram per ml and rifampicin selection is 10 microgram per ml. So accordingly, rif and can were added and a single colony from the agrobacterium plate is inoculated into the 5 ml broth. And this is cultured at 28 degrees Celsius, shaking for 2 days. Once the mother culture is ready, we prepare the working culture from the mother culture, where we inoculate 1% or 2% of the inoculum into LB broth. The volume can be 25 or 50 ml as required for your experiment. So what we try to do, we make 25 ml LB broth and we add the selection kanamycin and rifampicin, 50 microgram of kanamycin per ml and 10 microgram of rifampicin per ml, and then we add 2% or 1% inoculum from the mother culture that was raised. So this we culture for another 1 day so that the OD reaches 1 to 1.2, and then we pellet down the cells. The pelleting can be done at 5000 rpm for 10 minutes or 5 minutes, or 10,000 rpm 1 minute, preferably at 4 degrees Celsius. Once the pellet is prepared, the pellet is washed a couple of times using MS broth in order to remove the antibiotics, the residual antibiotics, dead cells and other floating debris. So once we prepare the agrobacterium, then we discard the soup and resuspend the pellet in 10 to 15 ml of MS broth. We add the phenolic acetosyringone, approximately 150 micromolar, to this particular MS resuspended cells, and this is the culture that we use for infecting the onion epidermal cells. Acetosyringone is a phenolic compound. It acts as a chemotactic agent which initiates the agro infection process. So the preparation of onion epidermal cells is after surface sterilization. The onion scales are cut and the epidermal cells are removed from this particular scales. The removed onion epidermis is then later incubated in MS broth or media. It can be for 1 hour or it can be even overnight. So these cells which are pre-incubated in the MS broth or media are then agro-infected with the agrobacterium that we have prepared containing acetosyringone. The incubation time is 1 hour. We incubate the cells or infect the cells in agrobacterium for an hour, or it can be even extended based on the requirement of the cells. Once the agrobacterium is infected, we transfer that particular agrobacterium, we rinse it in sterile water or MS broth, and then we co-cultivate this particular infected onion epidermal tissues. So for co-cultivation, we prepare LB plates or MS plates, preferably MS plates with the required salt, sugar and hormones you don't have to even add hormones. What we do, we pull out the onion epidermal cells, we rinse the onion epidermal cells that were infected using agrobacterium, and place the onion epidermal cells on tissue, sterile tissue, to remove excess moisture, and then we place it on the MS plates. This we incubate from 24 hours to 48 hours in dark at 28 degrees Celsius. So this co-cultivation is the time where agrobacterium infects the plant cell, transfers the tDNA, and integrates it into the host chromosomal DNA. The time can be optimized, the co-cultivation time can be optimized based on the expression level that you see. So once the co-cultivated discs are ready, we pull out the co-cultivated discs from the plate, and then we visualize the expression of the gene or analyze the expression of the promoter on a fluorescent microscope. Here we see the expression of GFP. The first panel is the control panel, where we have three categories of photo pictures. One is the bright field, the second is the fluorescent image for GFP, and the third is the merged image of the bright field and the fluorescence. The control, you can see no fluorescence coming, and the merged image also is certainly clear, whereas in the infected epidermal cells you can see clearly that there is good amount of GFP expression in the onion epidermal cells. So this can be used to study gene expression studies, promoter analysis, and for transient assays of important proteins. Thank you for being with me. I'll meet you again with another lecture. Thank you.